morning, good afternoon, good evening, depending on where you are in the world. It's an absolute pleasure for me to moderate this latest keynote series or keynote in a series for the Hamlyn Symposium on Medical Robotics. And today, I'm delighted to introduce a friend and colleague and a compatriot, Pietro, from the University of Leeds. So Pietro is professor of, is the chair of Robotics and Autonomous Systems at the University of Leeds, where he directs the STORM Lab, which focuses on intelligent robots to fight cancer, the Institute of Robotics and Autonomous Systems and Sensing, and the Robotics at Leeds network. On completion of his degree in electrical engineering from the University of Pisa in 2001, and after his PhD in biomedical engineering from the world-renowned Scuola Superiore Sant'Anna in 2006, Pietro secured an assistant professorship in the same institution, working with the team of Paolo Dario, before he moved to Vanderbilt in 2011, where he continued to flourish as an academic, focusing on surgical robotics before his move to Leeds in 2016. Pietro is widely published, has secured extensive funding both nationally and internationally. He serves on multiple committees and boards, notably with his senior editorship in medical and rehabilitation robotics for the journal IEEE RAL. And his work features in the news all the time. And to top it all, he's a successful entrepreneur with his company Win Medical, which he co-founded in 2009 and was acquired by a larger enterprise in 2017. Today, I'm delighted to introduce his talk entitled Medical Capsule Robots, A Fantastic Voyage, which I'm very much looking forward to. Pietro, over to you. You can share your screen. Thanks.
Thank you very much, Ferdinando, for the nice introduction. Can you see my screen? Yeah. So welcome to everyone listening, and a big thanks to Ferdinando and the organizing committee for inviting me to share with you an exciting 20-year long journey in the field of medical capsule robot. Before I begin, however, let me warn you that this presentation contains scenes from preclinical experiments showing tissue, blood, and other things that some viewers may find disturbing. So viewer discretion is advised. When I was an early career researcher, I was always fascinated by talks given by Paolo Dario from Scuola Superiore Sant'Anna. He was my PhD supervisor and a pioneer of robotic endoscopy. He often mentioned science fiction as a source of inspiration, and in particular, he was used to refer to a 1966 movie called Fantastic Voyage, where a submarine crew are shrunk to microscopic size and venture into the body of an injured scientist to repair damage to his brain. Another example along the same line is this episode of The Jetsons, aired in 1962, where George Jetson goes in for a physical examination and the doctor uses a special capsule called a peekaboo prober. Fast forward 38 years to the beginning of the millennium, and this vision became a reality thanks to Given Imaging, now part of Medtronic, who introduced into clinical practice the Pilcam. This is a wireless camera integrated into a capsule that, once swallowed by the patient, is able to travel throughout the whole gastrointestinal tract, propelled by natural peristalsis, while sending pictures to a set of body-worn antennas. This technology has been used by more than 3.5 million patients around the globe and is now the gold standard for the diagnosis of diseases in the small intestine. Of course, this technology also has limitations, and in particular, it is not able to interact with the surrounding tissue and carry out intervention. These limitations sparked the brand new field of research within the robotic community, medical capsule robots. As robotics may provide controlled locomotion, advanced sensing, interaction with the surrounding tissue to perform surgery, and many other functionalities. Over the last 2 decades, a large number of medical capsule robots were developed by different research labs addressing a variety of clinical needs and anatomical districts. For example, from drug delivery to random biopsy, from legged locomotion inside the colon to underwater navigation inside the stomach, from in situ histology to local delivery of surgical clips. In my presentation today, I will share with you my personal perspective on how the field has evolved in the past 20 years and trying to highlight what has been accomplished, what have failed, and what were the lessons learned. But first, let's discuss the clinical motivation. Depending on their size, capsule robots can be designed to operate in different districts of the human body. However, the most common application has been in the gastrointestinal tract due to the relatively easy access via natural orifices and the huge benefit they can provide there. From an anatomical perspective, the gastrointestinal tract is a long tube composed by the esophagus, the stomach, the small intestine, and the large intestine or colon. And it is home to different diseases, including cancer. Looking at 2020 cancer deaths worldwide, we notice that gastrointestinal cancers combined account to more than 2.2 million and are the main cause of cancer-related deaths. If detected early, however, cancer can be treated effectively. Here the example of colorectal cancer. As other cancer, it evolves in 5 to 10 years from an asymptomatic stage where simple treatment translates in a very high 5-year survival rate to a symptomatic stage where the 5-year survival rate drops dramatically and there are very limited options. For this reason, colorectal cancer screening is a common practice around the world, with screening programs starting as early as 45 in US and Australia, and 50 in the UK. The best way to screen for colorectal cancer is by colonoscopy, using a flexible endoscope. This is a 1.5 meter long tube with a camera and illumination at the tip, an irrigation channel to clean the camera or to insufflate the collapsed lumen, allowing for visualization. The tip can be articulated in 2 directions by cable steering, with the cables at the tip connected directly to 2 knobs controlled by the thumb of the operator. The flexible endoscope also has a channel to allow access for endoscopic instruments to interact with the tissue and collect biopsies for further lab examination. Or remove polyps, which are precursor of colorectal cancer. A very similar instrument is used to access the esophagus, the stomach, the small intestine from the mouth in upper gastrointestinal endoscopy. It is very interesting to note that the design of the instrument has not really evolved for the past 60 years. Since 1957, main progress has occurred in the visualization component, going from a bundle of optical fibers with the operator having to look through it, to ultra-high definition camera chips integrated at the tip of the instrument. All the rest has not changed. This out-of-date design is associated to all the downsides of flexible endoscopy. The first being pain. The flexible endoscope is designed to be introduced from its proximal end, and as such, the distal part needs to interact with the surrounding tissue to advance. So when a bend is approached, the instrument must, by design, stretch the tissue to progress forward. This causes pain in the patient and increases the risk of perforation. Then the user interface is highly unintuitive, with the thumb controlling 2 degrees of freedom and the other hand used to move the endoscope forward and backward. And finally, the instrument is extremely expensive by design with all its integrated components. So flexible endoscope must be reprocessed after each use, increasing the risk of cross-contamination and limiting the availability of procedures to center properly equipped with cleaning facilities. So medical capsule robots have the potential to provide a solution to all these problems, offering a painless and less invasive experience, using robotics to facilitate or even automate the procedure, and possibly reducing costs, enabling single-use devices and procedures that can be offered outside specialized facilities. So what are the components required for medical capsule robots to achieve these goals? They must possess proper actuators and mechanisms to interact with the surrounding anatomy and move around in a controllable fashion. They need sensors, including vision, to enable clinical diagnosis and other functionalities. And they need a brain, a CPU, communication with the outside world and the operator, and finally, they need power. All these need to be integrated in a limited volume. So size becomes the most important cross-cutting constraint. Typical target values for a capsule robot are a diameter of 11 millimeter and a length of 26 millimeter to allow for oral intake, or a little larger volume, typically 3 by 3 centimeter, for applications in the colon via retrograde access. Of all these components, probably the most interesting for our community is the part related to the actuation. So I will focus on this in the next slides, but I'm more than happy to discuss the others after the presentation. The first approach was to adopt onboard actuation by actively controlling the capsule using internal miniature locomotion mechanisms. Given the constraint in size, the most effective way to explore onboard actuation was to focus on the specific needs of the different anatomical districts. For example, the esophagus is a tubular cavity that is relatively easy to access. There, the main need is to slow down or even stop the passage of a capsule robot, which would otherwise take few seconds to reach the stomach. The stomach is a large collapsed cavity that needs three-dimensional motion capabilities. The small intestine is a long tubular cavity that is pretty difficult to access as it is located in the middle of the gastrointestinal tract. And its diameter is about 1 centimeter and is the same as a given imaging Pilcam. Finally, the colon is a tubular collapsed cavity about 3 times larger than the small intestine. And there, a medical capsule robot needs to create space, move forward and backward with a reasonable speed, and have the possibility to interact with the tissue to get samples and remove lesions. Now, for the sake of time, I will only focus on stomach and colon, where the incidence of cancer is the highest. It is also worth mentioning that the small intestine is served very well by the clinically available wireless capsule endoscope, like the Pilcam, and the esophagus is extremely easy to reach via flexible endoscopy. As for the stomach, the first issue is to create space. While gas insufflation is typically used in flexible endoscopy, an alternative is asking the patient to drink about 1 liter of clear liquid to expand the stomach. To support this approach, within the European project Vector, running from 2006 to 2011, and coordinated by Mark Schur at Novineon and Paolo Dario at Scuola Superiore Sant'Anna, a volunteer drank a liter of water and then swallowed a Pilcam. The volunteer was then asked to assume different positions to allow the capsule to visualize different anatomical landmarks in the stomach, demonstrating images of acceptable quality to perform a diagnosis. This underpinned the development back in 2009 of a 3D printed 4-propeller miniature submarine, integrating 4 DC motors, a lithium-ion polymer battery, and a wireless microcontroller within a diameter of 11 millimeter and a length of less than 3 centimeter, with additional space to accommodate a camera. Here you can see different videos demonstrating the technology, with the capsule performing figure-8 trajectories at different speeds and dexterity trials. While this prototype was battery operated, thus with a limited lifetime, we also explored, in collaboration with Bob Puer's group in Leuven, wireless powering. We did so by integrating a set of 3 perpendicular coils and transferring power with a carrier at 1 megahertz from an external coil to be placed around the patient. In this experiment, we were able to transfer an average power of 400 milliwatt and operate successfully all the 4 propellers, all within the safety limits allowed for human exposure to electromagnetic fields. Well, after demonstrating the feasibility of this approach, we did not pursue it further. It is exciting to see a recent announcement from a Silicon Valley startup that is trying to bring a 4-propeller-based capsule robot to the patient for non-invasive inspection of the stomach. The company is called Indiatics and have been successful in demonstrating the technology in humans in 2020. You can even find a video on YouTube with one of the founders swallowing the capsule robot and streaming images directly from his stomach. So the idea of swallowing a small submarine to get a diagnostic test may soon become a reality. But now let's go back and focus on the colon. There, an intriguing way to create space and at the same time propel the capsule robot forward and backward has been legged locomotion. Pioneered by Paolo Dario and Arianna Menciassi's group at Scuola Superiore Sant'Anna in the first decade of the millennium via multiple projects, including Hemilock, Optimus, and Vector. Prototypes of increasing complexity were developed using shape memory alloy to actuate, shape memory alloy actuation at first, and then moving to miniaturized electromagnetic brushless motors. The number of legs increased from 4 plus a balloon, to 8, and finally to 12, to provide a more uniform lumen distension and a more efficient propulsion. In this version, 2 brushless motors controlled independently 2 sets of 6 legs. Depending on the gate of actuation of the motors, this capsule was able to propel either forward or backward at a speed of 5 centimeters per minute, generating a pulling force of 3.8 Newton. While this robot was an interesting example of mechanical design and miniaturization, legged locomotion was flawed by several limitations. The first was complexity. The capsule was made out of 72 miniature components, some requiring multiple technologies for their fabrication. As you may imagine, assembly was not that easy either. This meant a very high cost of production, thus a very unlikely translation into a cost-effective product. The second limitation was related to power requirements. In order to complete its journey along a colon of average length, the capsule required a battery pack that was about the same size of the capsule itself. And this was clearly unacceptable from a clinical standpoint, as ingesting 2 interconnected capsules would pose a high risk of retention. So due to complexity, powering, and size constraint, we had to abandon legged locomotion and get back to square one in search for a more feasible actuation strategy. Then, while scratching our heads in lengthy brainstorming meetings back in 2009, we came across this science fiction clip.
Using this magneto, I will now guide the keys up the thorax and out via the frontal face hole. That's the storage locker. A boat. The other boat.
And then the big idea, magnetic manipulation. So placing a permanent magnet inside the capsule robot would enable external magnetic field to generate forces that are related to the applied field gradient and torques that are related to the misalignment of the 2 fields. So by controlling the external magnetic field and field gradient, we are able to apply a controlled wrench to the capsule robot without the need for strong power or integrating complex mechanisms on board. While magnetic manipulation has been explored since 1950 to guide cardiovascular catheters with a magnet integrated at the tip, the first example of a magnetic capsule robot dates back 2008, when within the European project Vector and in collaboration with Ovesco, we designed the capsule for the controlled deployment of a surgical clip to stop bleedings in the gastrointestinal tract. This here is the over-the-scope clip developed by Ovesco, which is typically released by a flexible endoscope. And this is the capsule robot integrating a mechanism to release a clip when receiving a wireless command from outside the body of the patient. The release mechanism is actuated by a brushless motor integrated on board, and a permanent magnet is also included in the capsule, allowing for remote control and manipulation. In this in vivo experiment, the capsule robot is moved toward the bleeding via magnetic manipulation. Then, once the capsule is in position facing the bleeding, the operator sends the wireless command and the clip is released. And then the capsule is moved away still by magnetic manipulation. In this first example, the external permanent magnet was supported by a passive arm and manipulated directly by the operator, often limiting the precision of motion. To improve this, we mounted the external permanent magnet on a robotic arm. As you can see from this video, using small cylindrical permanent magnets in the capsule enable a very good level of control over pan and tilt. Then we repeated the experiment using an in vivo animal model and compared robotic against manual control of the external magnet to manipulate the capsule inside the colon. The results were clearly in favor of robotic control, so we decided to adopt this strategy in all our research platforms. Of course, using a permanent magnet outside the patient is not the only way to generate the driving magnetic field. Another possibility is given by electromagnets, as in several solutions proposed for steering the tip of cardiovascular catheters, as well as for manipulating endoscopic capsules. Electromagnets provide an additional degree of control in varying the magnitude of the magnetic field by changing the current intensity in the coils. On the other hand, permanent magnets offer a higher volumetric magnetic flux density, typically resulting in a more compact driving system. Another interesting distinction is how to generate motion. While we have been focusing on dragging the capsule via gradient control, another possibility is to use rotating fields to advance the capsule with spiral threads rotating around its main axis. If you want to probe further on magnetically guided capsule endoscopy, then I suggest this interesting review paper appeared in Medical Physics. Now that we have discussed how magnetic actuation can enable navigation, let me give you a few examples of magnetic capsule robots that are able to perform specific functions. The first example is a biopsy capsule. The goal here is to get random biopsies in the small intestine to monitor Crohn or inflammatory bowel disease. To do this, we designed a capsule composed by two permanent magnets located at the far ends, and a central unit that is free to rotate around the main axis and a lateral opening. The central unit is composed by a semi-circular blade and another two permanent magnets working as a magnetic torsional spring. Where no external magnetic field is applied, the two magnets in the central unit rotate to expose the opposite pole to the magnets next to them, as you can see here. In this way, the lateral opening is closed by the back of the blade. Then, if an external magnet is brought forward, the biopsy capsule is attracted to the tissue due to the interaction with the lateral magnets. At the same time, the central unit rotates under the effect of the external magnetic field, loading the magnetic torsional spring and opening the capsule. Once the external magnet is removed, the central unit rotates back by 180 degrees under the effect of the magnetic spring, with the blade cutting the tissue and storing it inside the capsule. Here you can see a clip of the capsule in action, with opening and closing of the hole as the external magnet is moved in and out. Then an experiment inside explanted porcine tissue showing how the capsule is able to collect a biopsy sample successfully. It is worth mentioning that we do not have any electronically powered component in this capsule. This allowed us to achieve extreme miniaturization and to avoid limitations due to battery lifetime. Another brilliant example of smart design is the magnetic actuated soft capsule endoscope by Metin Sittis group. This is the first example of a capsule mixing magnetic actuation with soft robotics. And also in this case, we have a bistable magnetic mechanism. When the external field is below a certain intensity, the capsule can be moved around by rolling locomotion. Then, when the operator needs to trigger a certain function, like drug delivery, the intensity of the external magnetic field can be increased to a level that aligns the capsule first, and then overcomes the resistance offered by the four soft hinges to the reciprocal attraction of the internal magnets. While the first prototype, for the first papers, this capsule was proposed for drug delivery, more recent works demonstrated needle biopsy sampling and multi-scale robotic approach, where the main capsule delivered tetherless microgrippers aimed at grasping small samples of tissue, and then recovered them via a wet adhesive patch that you can see here. Another interesting example, this time mixing magnetic actuation with origami design, is a capsule robot for patching stomach wounds, designed by Shuei Miyashita. In this case, a biocompatible patch is folded around the magnet and frozen in water. As the capsule is ingested and the ice melts, the patch unfolds, providing a structure for the robot to move around via magnetic manipulation, and a biocompatible surface to patch a wound. So another example of a magneto-mechanical design with no electronics on board. Now, let's have a look at how magnetic actuation can be used effectively in different regions of the gastrointestinal tract, and in particular in the stomach and in the colon. A key element for magnetic actuation is the availability of space to move in. In the stomach, as we discussed previously, space can be created by ingesting water. Following this approach, early experiences started with manual operation of the external magnet, then moved to modified MRI machines to control the external field. And finally, the field settled on a system where a permanent magnet is controlled by a robotic arm, as in the platform developed by Ancon in China and commercialized by ANX outside China. This platform has recently been approved by the FDA for gastroscopy and is available in hospitals in China and the US. It is interesting to mention that the system works in open loop without tracking the pose of the capsule and assuming that there is enough space in the stomach for the capsule to align to the external field. And we will get back to this point in a few slides. Now let's focus on the colon. Typically, the lumen is collapsed, as you can see here, and magnetic manipulation is not effective in a collapsed lumen. As demonstrated in this video. Also, the colon is permeable to water, so the same approach used in the stomach does not work here. So following the steer from our clinical advisors in the Vector project, we decided to go for retrograde access, drop the wireless approach, and have a soft tether to provide gas insufflation. In addition to that, the tether provides electrical connection to the electronics and access for conventional endoscopic instruments. This design, that we have called magnetic flexible endoscope, provides all the functionalities of a conventional flexible endoscope, including the possibility to get biopsies and remove polyps. Differently from conventional endoscopes, the force to advance in the colon is applied at the tip of the instrument, pursuing a sort of front-wheel drive approach that drastically reduces the force required to progress forward. So the body of the endoscope becomes extremely flexible, and this should avoid stretching the surrounding tissue, prevent pain, and eliminate the risk of perforation. Another advantage is that thanks to robotics, the procedure may become more intuitive for the operator, thus reducing the long training required to gastroenterologists. And finally, the device itself becomes extremely simple, with no moving parts and a low cost of fabrication, opening to the possibility of a single-use device. So this design has the potential to address all the main limitations we discussed earlier about colonoscopy. Of course, this is not the only robotic colonoscope that has been proposed. Other notable examples include inchworm locomotion, a crawler based on micropillar threads, and more recently, the idea of a self-growing soft pneumatic eversion robot. For a complete review of the field, I encourage you to have a look at this review paper appeared in the Journal of Clinical Medicine. But let's get back to magnetic flexible endoscopy. In this study, performed in 2013 by Alberto Arezzo from the University of Turin, we compare robotic with conventional colonoscopy in an anatomical simulator of the lower gastrointestinal tract. To simulate lesions, pins were placed randomly and blindly to the operator. The user controlled the magnet mounted at the end effector of the robot with a joystick while looking at the image stream coming from the magnetic endoscope. These results showed that the robotic approach was comparable to conventional colonoscopy in successfully reaching the end of the colon and in the number of lesions detected. However, the robotic procedure was 3 times lower, which is not acceptable from a clinical perspective. The reason for this disappointing result was related to the assumption that the magnetic endoscope was free to move in the insufflated colon following the motion of the external magnet. This, however, was not always true, as sometimes the tip remained trapped into tissue folds or the tether hampered the motion of the tip in a certain direction. Since the operator was not able to recognize this immediately, the magnetic link was often lost due to the external magnet being moved away from the tip of the trapped endoscope. In these instances, the operator had to retrace the previous step, try to re-engage the magnetic link, and move in a different direction. This was extremely time-consuming and frustrating for the operator and clearly underlined the need for real-time localization of the magnet inside the body of the patient. This crucial need is clearly highlighted in this work by J. Cabot's group, proposing closed-loop control of a magnetic capsule floating in liquid. Considering that magnetic field and field gradient are highly nonlinear in space, real-time tracking of position and orientation, or pose, of the permanent magnet inside the body of the patient is crucial to close the control loop. In this case, pose tracking was obtained by using 2 perpendicular cameras, observing the capsule through the clear liquid. To apply a similar approach to patients, we need a pose tracking strategy that is able to localize the device inside the human body and that is not affected by the magnetic field used for actuation. This excludes all solutions based on electromagnetic tracking, as coiled sensors used there are subjected to saturation when exposed to the high magnetic fields. So we developed our own real-time pose detection strategy by integrating 1 accelerometer and 3 couples of Hall-effect sensors inside the tip of the magnetic endoscope. The Hall-effect sensors are aligned along the 3 main Cartesian axes and placed around the internal magnet. Once the offset due to the internal magnet is removed, the Hall-effect sensor provides a reading of the magnetic field vector generated by the external magnet at the tip of the magnetic endoscope. This vector is then rotated by the angles provided by the accelerometer, used as an inclinometer, and then mapped in the same reference frame as the external magnet, whose position is known. Then we perform a search in a pre-calculated magnetic field map that gives us the position that the tip should have to read these values for the external field. Since the external magnet is a cylinder, magnetized along its main axis, its field presents an axial symmetry. This greatly reduces the search space, allowing the pose tracking algorithm to run at 1 kHz. Once the pose of the internal magnet is known, it is straightforward to estimate the intermagnetic force, which is also the force that the internal magnet is exerting on tissue in the direction of the external magnet. So monitoring this force in real time is extremely important to make sure that it always stays below the threshold that would cause a damage to the tissue. This method was proven successfully in the target workspace, with the only exception of a singularity plane in the center of the external magnet and perpendicular to its main axis. There, the magnetic field is always the same, and the position of the magnetic endoscope cannot be discriminated. For this reason, we had to introduce a coil perpendicular to the external permanent magnet, as you can see here. And this coil creates a field that is different at any point in the singularity plane, allowing us to identify the position of the magnetic endoscope. In this video, you can appreciate how the estimated pose in red is close to the true one, as measured by the encoders of the robotic arm moving it around in this experiment. Of course, this is not the only possible localization strategy. There are others where sensors are worn by the patient or integrated inside the surgical bed, and others specifically designed for magnetic rolling. These are all described in this recent review paper on localization strategy for medical capsule robot, appear in expert review of medical devices. Now that we have discussed how to manipulate the capsule robot via magnetic field and track its pose and force in real time, the next challenge is to identify the right level of computer assistance that would make the procedure intuitive to perform for the operator. Exploring level of computer assistance is a hot research topic in robotic surgery. Drawing inspiration from the field of automotive, this well-known editorial appeared in 2017 in Science Robotics, defined the number of levels of increasing autonomy in the field of medical robotics. We basically go from a level of no autonomy, where the robot is transparently teleoperated by the surgeon, like the Da Vinci surgical robot from Intuitive, to increased level of computer assistance that would enable, for example, autonomous execution of tasks like suturing, and autonomous completions of parts of the surgical procedure. In our research, we have used magnetic manipulation and real-time pose and force tracking to explore how these different levels apply to robotic colonoscopy. The first level of autonomy is robot assistance. In this case, the user is controlling the tip of the endoscope with the joystick by looking directly at the endoscopic image stream. Forward, backward, left, and right are related to the reference frame of the endoscopic camera, transforming the user experience in something similar to driving a car in a video game. The system computes in real time the position and orientation of the endoscope tip and decides how to best move the external magnet to achieve the motion desired by the user. Results from a usability trial involving 10 non-expert users, each repeating a colonoscopy in a training simulator for 5 times, showed a completion rate of 100% and a completion time of less than 4 minutes, which is comparable with conventional colonoscopy. A second level is task autonomy. In this case, we automated the task of retroflexion to allow the gastroenterologist to angle the camera at 180 degrees and look behind tissue folds. This is very important because often polyps may be hidden behind tissue folds, as you can see here. A conventional colonoscope can only retroflex in the first part of the colon by being pushed against the wall of the rectum. Thanks to its enhanced flexibility, our magnetic endoscope can retroflex everywhere in the colon. However, deciding how to move the external magnet to achieve a 180-degree turn is not trivial for a human operator. Therefore, we automated the task and developed an algorithm that acquires the pose of the endoscope tip in real time and computes the best trajectory for the external magnet to achieve retroflexion. In a work appeared in gastroenterology, we successfully demonstrated this approach in a porcine animal model, showing an average time required for retroflexion of 11 seconds. Then we automated another task, tip levitation. So in order to reduce the time that the tip is in contact with the tissue, we closed the control loop in force, always applying the right amount that would keep the tip of the endoscope levitating in the center of the lumen. This reduced the contact between the tip and the wall from 100% to 28% of the time. This approach was improved further by applying dynamic adaptive backstepping to better reject external disturbances and enable us to perform full colonoscopies in levitation mode in training simulators as shown in this video. Another task that can be automated is ultrasound scanning. Magnetic manipulation always forces the device inside the body to stay in contact with the tissue. This is ideal when working with ultrasound, where contact with tissue is required to guarantee a proper signal. The challenge, however, is how to control the intracorporeal probe to maintain a stable contact during an ultrasound scan and how to reconstruct meaningful ultrasound images. In collaboration with the University of Glasgow, we modified the tip of our magnetic endoscope to accommodate 2 micro-ultrasound transducers, able to acquire images of the tissue with millimeter scale resolution to provide in situ histology, and decide whether a certain lesion is malignant or not, without the need for sending a biopsy sample to a pathology lab for further examination. So a first step for us was to define a parameter from the ultrasound signal that quantifies the quality of the echo received. Whenever this parameter was above a certain threshold, the signal was considered of good quality, as you can see here. The robotic platform was programmed to manipulate the probe against the target tissue until the echo signal was strong, and then maintain a similar strength while rejecting external disturbances. When performing a scan, first the platform established a strong echo and then started a sweep in the desired direction, always trying to maintain a strong echo. The signal was then reconstructed by taking advantage of our real-time localization. Here we see the ultrasound image of a phantom with embedded features. Once we were satisfied with bench trials, we moved to a porcine animal model for validation. First, we reached the desired position in the column by magnetic navigation, and then we tried to acquire a micro-ultrasound image via manual teleoperation of the external magnet with scarce results. Then we tried the autonomous routine based on real-time localization and ultrasound signal strength. And we were able to acquire a clear image of the mucosa layer, demonstrating for the first time how intelligent magnetic manipulation can enable the acquisition of micro-ultrasound images from regions deep inside the body. And then finally, the highest level of autonomy that we can reach, autonomous navigation. In this case, the platform autonomously recognizes the direction where to go by identifying the lumen from the endoscopic image. The green dot here represents the outcome of the lumen detection algorithm running in real time. Then the platform drives the tip of the endoscope in that direction by leveraging the information coming from the post-tracking algorithm. The only thing left to do for the user is to feed the tether. In recent benchtop trials involving 10 non-expert users performing 5 robotic colonoscopies each, we achieve an average time to reach the sequence of about 4 minutes and 100% rate of success. This opens up the possibility for the procedure to be performed by endoscopy nurses, thus widening the availability of colorectal cancer screening for patients. We then evaluated autonomous navigation in a pig animal model, whose anatomy is more convoluted than an average human patient. And we were able to navigate autonomously up to 85 centimeters. In this plot here, you can appreciate how convoluted and three-dimensional was the trajectory navigated by the magnetic endoscope, confirming the great potential of intelligent magnetic navigation of capsule robots in the colon. Now, let me close my presentation with a summary of this journey and an highlight of some of the lessons that we have learned along the way. We started from year 2000 with the introduction of wireless capsule endoscopy by given imaging. This disruptive innovation motivated the robotic community to start investigating medical capsule robots, with initial efforts aiming at providing active locomotion by integrating miniature actuators and mechanisms inside the capsule. This approach struggled to provide clinically relevant solutions due to size and power constraints, so the focus of research shifted to magnetic actuation, requiring only a small permanent magnet inside the capsule robot to work. From a research angle, magnetic actuation was combined with smart mechanisms, soft robotics, and origami design. While from a clinical perspective, it reached the patients with the Ancon Navicam platform for investigation of the stomach, and is now available in hospitals. To address the relevant clinical need of improving colonoscopy, so in the colon, the idea of a wireless capsule robot was abandoned in favor of a soft tethered approach, as magnetic manipulation needs insufflation to work in the colon. In the middle of the last decade, we realized how real-time post-detection was crucial to enable closed-loop control of a magnetic capsule robot in an unstructured environment, such as the colon. Real-time post-tracking enabled us to explore different levels of computer assistance in robotic colonoscopy, and brought us to demonstrate the feasibility of teleoperated and autonomous colonoscopy with completion times that are comparable to conventional colonoscopy. The next step for us is now to transition to first-in-human trials, thanks to the support that we have secured from Cancer Research UK and the NIH. And this brings me to my last remark. As you may have noticed, research impact takes time to materialize, especially in case of clinical application. So perseverance is extremely important. To our young audience today, don't give up too easily. And on this note, I'd like to conclude by thanking who has been with me throughout this journey. First of all, my mentors, Paolo Dario and Arianna Menciassi, the whole Vector team, including Alberto Arezzo, Mark Schur, and Mark Schur, who gave us a strong clinical steer from the early days, and the fantastic research team at Scuola Superiore Sant'Anna, where I've been until 2011, and in particular, Gastone Sciuti and Massimiliano Simi. Then my group and collaborators at Vanderbilt University and the Vanderbilt Institute for Surgery and Engineering, where I was from 2011 to 2016. Here, a particular mention to Keith Obstin, who remains one of the most committed clinical champions of magnetic colonoscopy. And then my current team at the University of Leeds. And last but not least, the general sponsor from the two sides of the Atlantic that made our research possible. And with this, I thank you for your attention. I'll be more than glad to take questions.
That was wonderful. Pietro, what a fantastic presentation. Thank you very much.
Thank you.
And thank you on behalf of the audience. Now we have a few minutes for questions. I'm just reminding everyone, if you would like to pose a question to Pietro, please put your question in Q&A, and I will read it out for Pietro to answer. And I've got the first one for Mel, who says, great talk. Thank you for this perspective. When is the first in-human plan for this new technology?
So we are now in putting together all the paperwork required in the UK by MHRA to clear us for starting first human trial. And we are hopeful to get clearance after the summer in autumn and start the first human trial by the end of the year or early next year. And then in parallel, we have also secured support from the American NIH to perform these trials also at Vanderbilt University in the US. And then the first trial are planned next year.
So Pietro, and with the NIH connection, how complicated is the process because the research was done in the UK, or is because you have a double affiliation there that it actually makes it easier for you to execute the trial through that?
That's a very good question. So as we are now, we are working in the US, we are working directly with the local institutional review board. So since colonoscopy is a class 2 device, and we have done all the work to make it extremely safe, we are hopeful to convince the IRB at Vanderbilt University that our platform does not pose significant risks. If we are able to convince this, they will be happy for us to start the trial. If we are not able to convince them, then they will ask us to go to the FDA, get from them the non-significant risk determination, and then go back to Vanderbilt. So the process may be as simple as just convincing the local IRB. In the UK, is similar to the other option. So we need to go to the national body to get clearance.
Just start a vlog though, or a blog right? So that you basically try to record some of that experience, because it's hard enough to do it through one ERB, through one institution in the UK. And the fact that if you are able to do it concurrently in the US and the UK, I think it would be a great case study for people to learn from. We've got plenty of other questions. I've got the next one from an anonymous attendee, and he says, or she says, what are the materials used to make the capsules? How do biodegradable robots exactly work? And where does the magnet go?
So the magnet goes inside, and permanent magnets are typically not very biocompatible, but as long as they are encapsulated in a fail-safe and biocompatible material, that is fine. Typical biocompatible materials are plastic like PEEK, but biodegradable material are different. And in some cases, those are same material as jellies, like sugar jellies, that can be absorbed by the body. And so there is also, I've seen some work in edible robots. I think from EPFL, I've seen those. So robots that are composed by sugar gel, and once they have performed their action, they just dissolve. And of course, they can also integrate drugs.
And to clarify this person's question, so because of the fact that you're using magnetic actuation for the latest work that you presented, one element of the capsule is always in contact with the tissue, right? Is that correct?
That is correct. Although we also had an implementation where we made the tip levitating. So basically we had the magnet and we were pulsating very quickly the external magnet so that the tip was floating and basically the equilibrium was with the tether.
But because you got opposing forces, right? You got magnetic and gravity, or
yes.
I don't know,
yes.
first self-is one of those. Okay, I get it. So there's a question from Andy Wood here, with actually one that I was also curious about. So what is it that Pilcams cannot do with a classical laparoscope can do, which may explain why, even though they exist as a commercial entity, we really, the common Joe and Jane has never seen one, and it's not the sort of thing that is widely adopted in clinics and hospitals.
So the main limitation of the Pilcam is that you cannot get biopsy samples, and you cannot remove the polyp. So in especially colon cancer, it develops in time. And so if you see a polyp, and you can remove it straight away, and in that place you will never develop cancer anymore. So it's extremely important, if you see something, remove it. And that is something that the Pilcam can only see, and then find it back is extremely difficult. So that's why Pilcam colon never really is indicated only in case a colonoscopy fails because of the anatomy. Then you get a Pilcam
because
colon.
it would still then be so it saves nothing, right? Because it would have to be a two-stage process as well. And so what is the most natural application for which Pilcams work today?
The small intestine. So the small intestine is in between the stomach and the colon. There, typically, cancer does not develop with polyps. There are no polyps, really. So there is mostly celiac disease and inflammatory bowel disease. So this can be diagnosed with the Pilcam, and treatment does not involve surgery. It's more like medications and diet. So small intestine is basically the gold standard is Pilcam.
Fantastic. Thank you, Pietro. Plenty of questions here. So Xiao Guang Dong, yeah, they ask, thanks for the great talk. In addition to the GI tract, how do you envision that magnetic endoscope robots could be further developed for medical functions in the vascular structures with fluid flows and other organs such as the brain, lungs, etc.?
So in that case, I'll answer with the research that we are doing in my lab is trying to work on magnetic tentacles. So instead of just controlling one point, we would like to control the full shape of a continuum robot or a catheter. because of course the catheter, like a flexible endoscope, is introduced from the distal end, and so to advance needs to stretch the tissue. And so what we are trying to do is to control the entire shape by magnetic fields, so that this traumatic interaction with the surrounding tissue doesn't happen anymore. As well as we can also implement like follow the leader motion around obstacle for brain surgery. It's very preliminary research. We started with the ERC Consolidator Grant No Limits in 2019. We are starting to have first results next talk.
Fantastic. And we'll look forward to having you then. Pietro, there's another question that I think tallies with one that I have myself, which is, they ask, and this is Andy Wu, what is the biggest difficulty in capsule robot to go to hospital? And I think I'd want to follow that with, what do you think are the stumbling blocks to get this into a product and out to patients? So you just gave us an outline of the first-in-man timeline. Now, what is the longer term, and do you have the appetite to spin out your own second company? Or is it something you'd rather put in the hands of someone else and then see it?
Yeah. So the field of gastroscopy, we have seen there is this robot magnetic manipulation platform developed in China, now available also in the US, without localization, as I mentioned before. That is in the hospital. But again, doesn't interact with tissue. For us, we are trying to we have a plan to spin out a company and try to move this forward once we see the outcome of the first in-human trials. Colonoscopy is a very specific field with a strong presence of companies like Olympus, with a very strong market, a very strong group of users. I think, and very low costs. So the cost of a colonoscopy is extremely low. So it's a challenging field. There have been a lot of startup, also robotic startup with robotic colonoscope, trying and failing. We hope that combining multiple values like no pain, ease of use, and ultra low cost, making the device disposable. And so riding the wave now of disposable flexible endoscope, the three combined will give us an edge, but it's tough.
Yeah. So to complete it, the $1 million question, is self-propulsion dead, or do you think it will make a comeback as we see evolution in other allied technologies, for example, brand new ways to increase the power density of batteries, for example?
Oh, that's a very good point. So you mean onboard locomotion, like?
Correct. Yeah, and this is actually not my question. This is an interpretation of a question from another anonymous attendee who's asking exactly this.
So of course, all this, like the given imaging, Pilcam in the year 2000 was driven by advances in miniaturization of cameras and extremely efficient wireless transmission of images. So driven by the technology advancement. And nowadays we are seeing a similar trend with batteries. So high capacity battery driven by the automotive, really. Yeah. So assuming that this technology evolves as fast as we expect, probably there may be extremely high power density batteries that we can use for capsule robots, extremely efficient actuators, maybe also better wireless powering. That would resurrect the field of onboard actuation for sure. Yeah.
Wonderful. Thank you, Pietro. This has been an amazing talk. We're out of time, so I just wanted to thank you once again for taking the time to give a wonderful keynote to our audience, and remind our audience that whoever has not been able to see this live can see a version of this talk available on www.hamlynsymposium.org or via our YouTube channel. And remind everyone there is a second keynote from Pierre Janin later this week, who I would all welcome to attend. And so thanks again, Pietro. Thanks to the audience. And thank
you, Ferdinando, for organizing it.
See you soon. Bye, everyone.
Bye-bye.